Bismillahirrahmanirrahim. Last time I talked about cell cycle and its regulatory mechanism. To understand healing process thoroughly, we must understand what are the various growth factors, what are the different signaling mechanisms in cell growth, and how the cells interact with their extracellular environment. First, we'll start with the growth factors. Proliferation of cells is mediated by polypeptides. These are known as the growth factors. Apart from their growth promoting effects, they are also important for cell survival, locomotion, contractility, and angiogenesis, etc. These growth factors act as ligands that binds to specific receptors, and thus they deliver signals to inside of the target cells. Once inside the cells, these signals through a series of molecular transduction, they will start transcription of genes, and thus these genes will be stimulated to perform different functions. This table shows different growth factors and their functions. For example, vascular endothelial growth factor, which is denoted by VEGF or VEGF. These are released by mesenchymal cells, and what does this VEGF do? It will increase the vascular permeability and acts as metogenic for endothelial cells. Students, this table is very important from your examination point of view, so you have to memorize it. Now, cell signaling and cell signaling mechanism. First, what is the cell signaling? The binding of a ligand to a cell receptor starts a cascade of events which will lead to expression of certain specific genes. Generally, there are three different modes of cell signaling. These are autocrine, paracrine, and endocrine signaling. In autocrine signaling, a cell signals to itself by releasing a hormone or a chemical agent that binds to receptors on its own surface. For example, in tumors, there is overproduction of growth factors and their receptors. These growth factors stimulate the unregulated and uncontrolled proliferation of the same cells as well as the adjacent non-tumor cells. In paracrine signaling, the signaling molecules released by a cell will only affect those target cells which are situated in its close proximity. For example, the conduction of a nerve impulse from one nerve cell to another or to a muscle cell. Endocrine signaling occurs when a cell releases hormone that acts on a distant target cell in the body, and they are usually carried by a bloodstream. For example, glands such as a thyroid gland and pituitary gland. These glands produces hormones and they are carried to distant sites in the body via bloodstream. Now, what is signal transduction? Signal transduction is a process by which a signal is converted from outside the cell and is taken inside the cell, and it will cause a functional change within that cell. There are three important stages of a signal transduction: reception, transduction, and response. On reception, a ligand will bind its specific receptor on a cell surface. This will result in conformational change in that receptor, which acts as a gateway. So this message will be taken into the cell cytoplasm through this gateway. Now, inside the cytoplasm, there are some other molecules which will decode this message, and this is known as the transduction. Once this message is decoded, it will be conveyed into the nucleus. Inside the nucleus, certain genes will start expressing themselves, and thus a cellular response will be generated. This figure shows main types of some cell surface receptors and their transduction pathways. Shown are receptors with the intrinsic tyrosine kinase activity, which is shown on extreme left side of this slide. 7 transmembrane G protein coupled receptors and receptor without tyrosine kinase activity. For example, VEGF will bind to receptor with tyrosine kinase activity, and through activation of the intracellular molecules, a cellular response will be generated. In case of tissue repair and regeneration, the interaction between cells and the components of extracellular matrix is very important. Extracellular matrix is constantly remodeling, and its synthesis and degradation accompanies morphogenesis, regeneration, wound healing, tumor invasion, and metastasis. Extracellular matrix perform a lot of functions. One of them is it sequesters water by providing turgor to soft tissues and minerals that give rigidity to bone. It will provide mechanical support for cell anchorage and cell migration. It will cause the control of cell growth, maintenance of the cell differentiation. It will provide scaffolding for tissue renewal. It will help in establishment of tissue microenvironments, and there will be storage and presentation of regulatory molecules within this extracellular molecules. Now, types of extracellular proteins. These include fibrostructural proteins, adhesive glycoproteins, and proteoglycans. Collagens and elastin, they come under the heading of the fibrostructural proteins. In adhesive glycoproteins, they connect the matrix elements to one another and to the cell. And the proteoglycans, hyaluronin, they will provide resilience and lubrication. So this figure shows some main components of extracellular matrix, which includes collagens, proteoglycans, and adhesive glycoproteins. Both epithelial and mesenchymal cells, they interact with extracellular matrix via integrins, shown here by a yellow star. Among extracellular matrix proteins, collagens are very important. In this table, different types of collagens along with their tissue distribution is shown. The last column shows the genetic disorders which are associated with these specific collagens. Elastin. Morphologically, elastin fiber consists of a central core of elastin, and it is surrounded by a meshwork of fibrillin. Any defect in this fibrillin meshwork will lead to skeletal abnormalities. For example, such abnormalities are seen in the Marfan Syndrome. Proteoglycans. They form highly hydrated compressible gels. They also play important role in cell proliferation, migration, and adhesion. The Hilar Runin, they forms a viscous gelatin-like material because they can bind water. Adhesive glycoproteins and adhesion receptors. These are the structurally diverse molecules which are involved in cell-to-cell adhesion, linkage between cells and extracellular matrix, and the binding between the extracellular matrix component. Fibronectin and laminin, they come under the heading of the adhesive glycoproteins, and adhesion receptors, which are also known as the cell adhesion molecules, CAMs. They are grouped into 4 families, which include immunoglobulins, adherins, selectins, and integrins. Fibronectin, these are of 2 types, tissue fibronectin and plasma fibronectin. They provide aggregates, or they form fibrillar aggregates at the wound healing site. Laminin, it is a most abundant glycoprotein in basement membrane. What does they do? They connect cells to the underlying matrix components, such as type 4 collagen and heparin sulfate. Integrins, they are a family of transmembrane glycoproteins. They are composed of alpha and beta chains. They are present in plasma membrane of almost all cells, but RBCs. So this figure shows there are molecules involved in cell-to-cell adhesion. In this figure, it is shown that how the extracellular matrix components and growth factors, They interact and activate these signaling pathways. For example, integrins bind the extracellular matrix component, which is shown here by a yellow arrow. And these integrins, how they interact with cytoskeleton at focal adhesion complexes, which is shown here by a red arrow. And thus the nuclear signals will be generated. Wound healing is a process by which skin or other body tissue repairs itself after trauma. In undamaged skin, the epidermis and dermis will form a protective barrier against the external environment. When that barrier is damaged or broken, a cascade of biochemical events will start and they will try to repair the damage. This whole cascade is divided into inflammation, angiogenesis, proliferation of fibroblast, scar formation, and connective tissue remodeling. Before we discuss the steps of healing and repair of cutaneous wounds, we should understand how blood vessels establish themselves during healing process. Angiogenesis is not only important from physiological aspects such as in wound healing, regeneration, etc., but also is a fundamental part of pathological process. For example, during tumor development, metastasis, inflammation, etc. When blood vessels are assembled during embryonic life, these are established either by angioblast, which are the endothelial precursor cells, or from hemangioblast. These are the dual hematopoietic and endothelial cell precursors. When a site is injured, inflammatory process will start and they will try to remove the injured tissue, and at the same time angiogenesis is stimulated. The formation of new blood vessels in adults is known as angiogenesis, and it occurs by two ways. One is by branching and extension of adjacent pre-existing blood vessels, or they can also be formed by recruitment of endothelial progenitor cells from bone marrow. Angiogenesis from pre-existing blood vessels, and this one is shows angiogenesis from the endothelial progenitor cells. Angiogenesis from pre-existing vessels. Whenever a stimuli is received, for example, in the form of hypoxia, a cascade of angiogenesis will commence. First, there will be vasodilatation and increased vascular permeability of the existing blood vessels. Then the extracellular matrix of these vessels will be degraded, and thus the endothelial cell will start migrating towards that angiogenic stimulus. Nitric oxide will cause vasodilatation, while VEGF will lead to increased permeability of these blood vessels. The basement membrane of the parent vessel will be degraded by matrix metalloproteinases, and the plasminogen activator will break down the junctions that hold the endothelial cells to one another. Then the endothelial cell will start migrating towards angiogenic stimulus. During this migratory process, there will also be proliferation of endothelial cells just behind the leading front of migrating endothelial cells. Once these endothelial cells reaches their destination, they will start maturing and remodeling themselves into capillary tubes. And then the further growth of these vessels will be inhibited. Finally, these capillary tubes will thus be organized into mature vessels by recruitment of periendothelial cells. This is a schematic representation of how the angiogenesis from pre-existing vessels occurs. Now, what are the growth factors and receptors involved in angiogenesis? Although there are a variety of factors that participate in angiogenesis, but VEGF is the Most important one. VEGF is secreted by many mesenchymal and stromal cells. There are certain specific receptors to which VEGF bind. All these receptors belong to a family of tyrosine kinase receptor. As I have already told you, VEGFR2 is the most important receptor for them. If any mutation occurs in this receptor, then there will be no vasculogenesis. The stimulation of VEGF is caused by hypoxia, platelet-derived growth factor, TGF alpha and TGF beta. So what causes the VEGF stimulation to start angiogenesis? Whenever oxygen level drops, then the VEGF are induced automatically and commencement of the angiogenesis. So this is a microscopic picture of granulation tissue showing numerous blood vessels, edema, and a loose extracellular matrix containing inflammatory cells. So the new vessels have formed. Now pericytes and smooth muscle cells will come, surround, and stabilize them. The angiopoietins 1 and 2 and platelet-derived growth factor and TGF-beta will help them in this stabilization process. There is a receptor on endothelial cell which are called as Ty2. The angiopoietin 1 will interact with Ty2 and they will recruit the periendothelial cells. In case of larger vessels, the smooth muscle cells are recruited by platelet-derived growth factor. TGF-beta they enhances production of the ECM proteins. So which ECM proteins or extracellular matrix proteins, they regulate the process of angiogenesis. These are integrins, thrombospondin 1, SPARC, and tenecin, proteinases such as the plasminogen activator and matrix metalloproteinases. All of these have their different function. So student, this is all for today. Thank you very much.